morning. Let's go ahead and get started. Thanks to those of you that showed up for the talk yesterday. It was nice to have it was pretty much full. I apologize for the sound system, which was embarrassing, actually. But so a lot of you had to get the information just from the slides that were written, and that's okay. If you don't understand some of those terms, like all of them, then you can Wikipedia that. That's a great way to get started on the topic, and even for your poster stuff too. What does that sound? Okay, yeah, they have another feature. Is that in here? No. Would you close that door and tell them to shut up? Or turn it off? No. Okay, so back to our goals. We've gotten through a couple of things. What we're doing is it's really boring to memorize a list of transport processes. But we have to get these players and understand how they work before we can put them together to make them do things that are important. So we're going to hit a lot of clinical applications today, and so that's where we're headed. I know some of this is kind of, it can be less than interesting. Okay. Now, one of the things we want to do also is construct a toolkit of inhibitors that we can use to probe which of these transport proteins are going on. Now, we talked about a drug yesterday called Wabane, which inhibits, and keep that closed, please. Thanks, appreciate it. It's not your fault. That inhibits the sodium pump, right? So that blocks it and it binds near the potassium binding site, shuts it down, okay? But there's one, is there one for the calcium ATPase? Absolutely, there is. It's called Vapsagargan. I didn't choose that name. It sounds like what's that movie with it's an alien robot, then it turns into a car, then into a dill pickle or something. Transformers, okay. This is a good, I've come to your planet to inhibit your circa. So, that Vapsagargan, okay. So you'll never forget that. So this blocks circa, okay, shuts it down. All right. And another one that I should have mentioned, I summarized these later in the notes, but let's go ahead and talk about them now, or mention them. Is there something that inhibits the GLUT family? Absolutely. It inhibits all of the members of the GLUT family, probably because it binds near the glucose binding pocket. It actually locks it into the inward-facing position so that it can't reverse and pick up a new set of cargo. So that's cytochalasin. And there's several cytochalasins, and this is B. So that inhibits all the GLUTs. GLUT 1, 2, 3, 4, 5. So those are useful things to have if you want to probe which of these proteins is doing their job in a particular epithelium. So you have a patient that has a particular problem, or you're studying a part of the kidney or something like that, you need to be able to test what's managing the transport. Okay, so done that. Where does that bring us? Let's go to the outline. And you want to be able to distinguish between these various types of pumps. So these are the three classes, and we talked about this. So that class for circa and sodium pump, those are the two poster children for the P class of ATPase family. And the P class indicates that it phosphorylates itself at some point in the transport cycle. Okay? That doesn't happen for any of these other pumps. Okay, so that's what's unique. The other thing is the actual transporter is a single polypeptide. Now, does it have quaternary structure as it sits in the membrane? Absolutely. The sodium pump, or at least the sodium pump circuit, is a single polypeptide, but the sodium pump is a heterotetramer. Okay, but the part that actually does the transport is the alpha subunit, and it is. Does it have multiple membrane-spanning alpha helices? All of these do. All transporters have multiple alpha helices that go across the membrane. So that's going to distinguish this class from this one. So these are called the F-type ATPase family after the singular member of that family, which is ATP synthase. This is what makes your ATP as long as you're breathing oxygen. Okay. And so this is a really interesting machine, but its formal name is called FO-F1 ATPase. The synonym is ATP synthase because you actually run the pump backwards and use a proton gradient to synthesize ATP. Instead of splitting ATP and then ejecting protons, it runs backwards. So we'll look at that. So this is the hallmark that makes it different from that is it's got 8 or more subunits, 8 or more polypeptides that fit together to do the job. The solute is exclusively protons that's moved. Okay, and where does it live? It lives in the inner mitochondrial membrane. All right, so let's take a look at this beast. And let's go to the PowerPoint. Okay, so here it is. Now, remember where you are. There's the inner membrane space. This is the inner mitochondrial membrane. The outer mitochondrial membrane is down here somewhere, right? And so remember, the inner membrane space is the exoplasmic compartment. The cytoplasmic compartment, there's 2 of them. Okay, it's the real cytoplasm, which is on the other side of the outer mitochondrial membrane. What have we talked about that's in the outer mitochondrial membrane? What protein? We've talked about one. It's our poster child for something. Beta baryl. Well, think about that. Okay, so this is another guy that's on a different part of that. This is the inside of the mitochondria. And remember, the inside of the mitochondria, the inner membrane, is extremely folded. And the reason it's folded is that it's standing room only for all the proteins that are doing their job. The other thing that's in this membrane, of course, is the electron transport chain. So what's happening is the food you eat that is taken into the mitochondria, and you blow off the carbon dioxide, you take those electrons, you pass them down the electron transport chain that's sitting right here, and the electron transport job is to eject protons into the inner membrane space. Why does it do that? Does it just want to acidify you? Absolutely not. A proton gradient is energy. And so what happens is those protons are going to go through this rotary machine, and it really is a rotary machine. When it goes through here, this little bell pepper thing is going to change its conformation in different ways and grab onto ADP and phosphate and slam them together and release ATP into your mitochondrial matrix. And then there are various transport proteins that take the ATP, because that doesn't do you a lot of good if the ATP is in the matrix of the mitochondria. It needs to be in your cytoplasm. Remember all those pumps we were talking about yesterday? Their ATP binding domain is facing the cytoplasm for a reason. That's because that ATP is made here and then ejected out. Okay? But let's go back to this guy. So it's got lots of subunits, but we can really simplify it if we split them up into 2 blocks. So the FO complex are all transmembrane proteins. Okay? And what is its function? It's a proton channel. So what it's going to do, instead of letting that proton gradient dissipate, just randomly float away, what it does is it grabs onto a proton, and then those protons then bind to this complex and start it spinning. So this thing rotates. If you're out in the inner membrane space and you're looking at the base of this, so your eye is down here and you're looking at that ring, it's going to rotate clockwise. And as it spins, there's a rotor right here that starts to turn. And what it does is it's turning inside of this bell pepper, and it touches it at 3 different places. And every time it touches it, it changes the conformation of that. This is the ATPase. So the F1 complex is where the ATP is actually made. So this is an enzyme that's making ATP. And so it alternates between 3 different kinetic states, and we'll talk about that when we talk about the mitochondria. But so what's happening is this thing is rotating, and that's providing the energy to make, or it's telling this bell pepper to change its conformation and make ATP. So this is the ATP machine, and this is the proton channel. So protons are going through here, and then eventually, after they go around the circle, they're ejected into the mitochondrial matrix, and then they can be shot across the membrane by your electron transport chain. So those 2 phenomena are coupled. And the reason we run this machine backwards is because we want to make ATP. Can we make this thing go backwards? In other words, can we make it split ATP, so make the bell pepper split ATP, and then eject protons. That experiment's been done. It is true. You can do that. You can incorporate this thing into lipid bilayers, and what you do is you put, you face it with a proton gradient where it's still lots of protons on the outside, very few on the inside. So the protons are trying to go across, but can't because it's facing the proton gradient. Well, if you give it ATP, this thing starts to spin because this thing is changing conformation, and it's causing the rotor to spin, and guess what? The protons are actually ejected. So if this thing splits ATP, the protons come out. If the protons go in, this thing makes ATP. So that's a really pretty neat machine, you have to admit. Okay. We'll go into the details of that when we hit the mitochondria a little bit later. Okay. Any questions about what's going on? Okay, and notice it doesn't violate our rule that the ATP binding domain of any protein that catches and binds ATP must be facing the cytoplasmic domain, because the matrix is the cytoplasm for what used to be a bacteria. Okay. That's exoplasmic, the outer membrane's here, and then this is the real eukaryotic cytoplasm out here. Okay. Now, there's another well-known member of this family, and it's shown there on the outline. Okay. And that's the VO-V1 ATPase. And so it's got the same look to it. It's got a rotary engine. It's got a proton pore. But its job is to run the way you expected ATPase to run. It splits ATP and ejects protons out of the cytoplasm or into a membrane. So, and so it's got the same, lots of subunits. The single solute is protons. And where does it live? It lives in the ER, vesicles, lysosomes, and on the plasma membrane. Okay. So how do you arrange it? And that's real easy because you know what the rule is. Okay. So if this is a cell, okay, and this, let's make a real big vesicle. That's not the nucleus. Let's say it's a vesicle. Okay. And we're gonna put this VO-V1 hump somewhere. Okay. And so you know that the bell pepper part has to face the cytoplasm. So you're going to put that bell pepper thing right here. Okay, that's the bell pepper. And then so that's the F1 group of subunits. There's multiple subunits. Okay. And then you're going to put the F, it's called the VV1, but it's in the F1 family. So it's V1 because we're talking, think vesicle or vacuole. That's what the V stands for. So the V1 is the bell pepper, and then you got to get the protons to go across the membrane, so you're putting a proton channel that spans the membrane. So what's going to happen is when this thing, so this is the VO group of subunits. And what you're going to do is you're going to split ATP by running it through the bell pepper. Okay. The bell pepper is going to change its shape, and it's going to spin this rotor, and it's going to take protons out of the cytoplasm and store them inside of the vesicle. And this is actually what makes the ER, the Golgi, and all vesicles specific, is this pump. The other neat thing is you can put it on the plasma membrane. How would you orient it? You use the same rules, correct? So this is the plasma membrane lipid bilayer. You're going to put the bell pepper here. So it's going to split ATP like that. The proton, so then the proton pore is there, ion channel basically, and you're going to take protons and eject them outside the cell into the exoplasmic compartment, just like the F1 pump could if it went backwards. This is actually, for this pump, this is actually the forward direction. Split ATP, eject protons. Can you make this, can you cause this to make ATP for you? Absolutely. If you give it a high concentration of protons, they'll flow through here and it'll take ADP and phosphate and stick them together and make ATP in the cytoplasm. It's kind of a cool machine. Okay. Questions about how that works? You see it's very different. Yes, sir.
Are you going ATP to ATP?
I'm sorry?
ATP to ADP.
When you split it,
Yeah.
you get two for the price of one, you get ADP plus phosphate. And I hope some of you saw the discussion on Piazza about the charge of phosphate and the charge, what is a protein, what's the charge of an amino acid become if it gets phosphorylated, because there's a couple errors in the book, so it's useful. So the charge on ADP is -3, the charge on, we never put it, because we just go, okay, you know that. But not everybody knows that ATP has 4 minus charges. 4. ADP has 3. And then when you phosphorylate a protein, amino acid like serine has no charge, correct? If you phosphorylate it, it now has -2 negative charge on it. Yeah.
Are there any structural differences between FO and BO?
There are, but they're real minor. So we kind of just go, well, they have the same 2 complexes, but there are some differences. Yeah. A lot of it's targeting and, yeah. Yeah.
Does the iron concentration have to be high in order for FOS1 to split ATP, or can it just switch?
Well, no, I mean, if you split ATP, you can drive the protons against the proton gradient. It'll go uphill, because you better be able to do that because you're using ATP, the energy of ATP hydrolysis to eject the proton. But if you capture the energy of letting a gradient dissipate, you can turn it into ATP. So that's one really core concept is energy gradients, proton and ion gradients are a form of stored energy that can be used to do stuff. Okay, good. Okay. Sure, I did my pointer. All right, so now let's shut this down. Any other questions? I think you're good. All right, so let's go into the last pump family, and this is a real weird one. It's a very important one medically. It's huge. This is the largest transport family that there is. There's 90 different genes in this family that make different kinds of what are called ABC transporters. Okay, and the hallmark of this group of transporters is this little cassette that actually splits the ATP. It's called an ATP binding cassette. And a cassette is just a recognizable series of amino acids that always have the same function. Okay? So obviously the function of this little cassette is to bind and split ATP. All right? So that's the character. So to be a member of this family, you have to have that ABC cassette. That's about the only thing that characterizes this family that's the same. It's really weird. Some are transporters, okay? They're classically ATP-driven pumps. Some of them are ATP-gated ion channels. Some are ion channels. The one that causes cystic fibrosis is an ion channel. It's a chloride channel. We'll talk about that. And some are not. They have this structure, but what they do is they regulate other ion channels, like the one in the pancreatic beta cell that's responding to glucose. That ABC transporter actually opens and closes a potassium channel. So it's just a regulator. This is a bizarre family, okay? What do they look like? So we're going to take the very first one that was discovered and look at its structure. Okay, and it's called MDR1. Okay, so here it is. And so the classic members of this family have 12 membrane-spanning alpha helices divided into 2 membrane domains. So they have one domain that starts, so the N-terminus is always in the cytoplasm. So where must the C-terminus be? Even number, same side, so the C-terminus has to be somewhere down here, right? In the cytoplasm, right? Okay, so it starts here, you get these big loops. Then this is an alpha helix, a transmembrane alpha helix, and it goes 1, 2, 3, 4, 5, 6. Then you go back to an intracellular loop. Why do you need to be on the back of the cytoplasm? Because this is where the nucleotide binding domain is. This is where it binds ATP. This is where the ABC cassette is located. ATP binding cassette is in that NB, nucleotide binding domain. And they just should say ATP binding domain, because it doesn't bind any other nucleotide except ATP. Okay, then it goes back in the membrane, 1, 2, 3, 4, 5, 6, and then it comes out, so it's always got membrane domain, cytoplasmic nucleotide binding domain, membrane domain, and finally it ends with a nucleotide binding domain. So it's got 2 engines. Okay. Now, so the reason it's called this multi-drug resistance protein, MDR1, is because its job in you, if you don't have cancer, is if you take a xenobiotic, that is a foreign drug of any kind, and it goes into your intestine, what this is going to do is the drug, they tend to be hydrophobic. The drug companies make them that way because they want it to get into the bloodstream and cross the GI system. Okay. What this thing does is as soon as it gets into, it passes this is the extracellular leaflet, exoplasmic leaflet, gets into the cytoplasmic leaflet, and this thing captures it and then splits ATP to eject it back into the GI lumen. And so you get rid of it. So this is a sort of a purifier, is what it's designed to be. There's some in your blood-brain barrier that keeps drugs out of your brain, and so that's really useful. So this is protecting you against all kinds of weird stuff. In order to be able to transport all of those things, it has to have one really interesting property. It's got a really sloppy affinity. This thing binds colchicine, steroids, aspirin, any kind of thing that loosely can be recognized as a xenobiotic. In fact, I like to call this the slut of the transport world. It will take anybody, okay? All right, so, and I say that not just to make you memorize it, but also people that have cancer often die because of this protein. So it really is an enemy, so I don't mind calling it a nasty name. But don't tell your parents to call it that. One group of people, and I hope you actually name your group, they called themselves the MDR1 sluts. That didn't go over too well, so that's already been taken. All right, so let's actually look at that problem. So let's get a little bit more knowledge about how it splits ATP, because this is the hallmark of this pump family. Okay, so the ATP binding cassette has all these amino acids, and it's divided into Walker A domain, a linker region, and a Walker B. And we're not going to worry about the linker, but Walker A and Walker B are very important because they're the 2 domains of the nucleotide binding domain that actually bind to ATP and pull it apart. And we know exactly which one does what. So the Walker A is here, and it's Walker A motif. It's holding on to the gamma phosphate of ATP. So here's ATP, right? And then there's the alpha, beta, and gamma phosphate. So one hand of this protein is going to grab that one, and then the Walker B motif binds magnesium, and magnesium binds to the alpha and beta phosphates. So it's doing this. And then when it splits it, it changes its conformation and pulls off that gamma phosphate, and you have ADP and phosphate. And it uses the free energy that's released from that to change the conformation of the protein and eject the drugs across it. Actually, one of my former collaborators and good friend of mine over at the Health Sciences Center in the Department of Cell Biology and Biochemistry, Ina Urbach, studies this protein. And so this is her model for how this thing works. And she actually studies both of the examples that I'm going to give you of this family. So what happens is some drug that you don't want comes in, intercalates into the membrane, and as soon as it gets into the cytoplasmic leaflet, sometimes they actually get into the cytoplasm, but it's actually going to be picked up when it gets there, okay? But the way this thing starts off is it's got the 2 membrane domains and the 2 nucleotide binding domains, and it starts off with the ATP already loaded in both nucleotide binding domains. They are loosely bound to both of these before anything happens. So what's the effect of the drug? The drug comes in here, and when it binds to the drug binding site, that causes the 2 nucleotide binding domains to rotate and dimerize. So they change and they face each other. They form a catalytic core, and one of those ATP fires. One fires. Okay. Now, it's not like the sodium pump, remember, which when it splits ATP, what does it do? What does it release? What? can't hear you.
Protons?
No, we're talking about the sodium pump. So it's splitting ATP and it releases something before it does anything with sodium. It releases ADP into the cytoplasm. This does not. It holds on actually to both ADP and phosphate. Remember, this is not a P-class transporter. This is a different class. It does not phosphorylate itself. The sodium pump phosphorylates itself on aspartate, but this one does not put the phosphate. It holds on to them, and it sequentially lets them go. And when it lets go of the phosphate is the first thing to be released back into where? The cytoplasm. That's what causes a conformation change that ejects the drug back into extracellular space. Okay? So see what you've done, you've vacuumed up the drug and you just put it back where it came from. And if you just keep doing this, you keep it out of yourself. Okay? And what happens to the ADP? You let the phosphate go, and then it sequentially next releases the ADP, and that's what returns it to this conformation to start all over again. So it only splits one ATP per cycle. Right? And the details of that sequence are written down for you here, so you don't have to write anything down extensively. Okay? Any questions about how that works? Think about how is it different from all the other classes. See, because you need to be able to sort these out. Yeah.
So it's only foreign drugs? Like,
well, it can do a few other things too. But this one specializes, well, if you can call being a slut, especially, it just transports all kinds of stuff. But it does actually, there are some that actually move phospholipids across membranes. And so that's why you have so many of them, because they all do really interesting things. Not all of them are transporting drugs. Now, so how does this kill a cancer patient? Well, this is why. So if you, particularly breast cancer is particularly bad because one of the most effective anti-tumor drugs in breast cancer is Taxol. And what Taxol does is it screws up the mitotic spindle so it can't work. And so what should happen is the cell should kill itself, correct? And so that's usually what happens. When it can't divide, it's just either going to be dead in the water or it will undergo apoptosis, all right? Now, so what happens is this is normally expressed in a lot of tissues, but what the tumor does is it overexpresses. So what you do is the oncologist is going to give this patient a large dose of xenobiotics, correct? Taxol is nasty, and it responds by trying to defend itself by inducing the synthesis and delivery of this to the plasma membrane. And so what happens is if you don't kill all of them on that first pass, this is what causes resistance to the chemotherapy. And what's really bad is it won't express just NDR1, it'll express usually these three different ones. They're all in the same family. So it doesn't matter, now if you try to go back and do another regime of chemotherapy, that woman's dead because there's no way that drug is ever going to get into her tumor until it reaches a concentration. Now you can jack up the concentration of those anti-tumor drugs. What's going to happen to the patient? The lethal dose is very close to the effective dose. And so they die. You can't give a drug dose high enough to defeat these. So people are working on new ways to destroy their effectiveness and all kinds of things. And a lot of new, let's see, I don't know if I show that or not, no. A lot of new specific anti-tumor drugs now are delivered because they bind to the receptor-mediated endocytosis machine that bypasses this thing. And so you can do an end run on it, you can inhibit it, so there's people working on all kinds of these things to get rid of this pump. And some of the genes that you're going to be isolating in Cell Lab are this family here. Okay. So what's really bad is if this thing is overexpressed, it's now resistant not only to the drug that you started with, it's resistant to all the drugs. Okay. That's why we call it the multi-drug resistance protein. Okay, any questions about that? And this is just one trick up a tumor sleeve. There's all kinds of resistance mechanisms. The key is to figure out what that person's tumor is doing to evade the drug and defeat it. And that's a pretty good strategy. All right, so let's talk about a second poster child in this family, and that's also medically important. And that's the thing that causes cystic fibrosis. And it has you don't call it by its name because its actual name is cystic fibrosis transmembrane conductance regulator. And if you want to call it that, that's fine. Impress your neighbors, but CFTR is the name we give it, CFTR. And as you can tell, it's a nice looking member of this family. It's got 2 membrane domains. It's got 2 nucleotide binding domains, but then it's got this weird little bulge, and that's not a tumor. That's a new domain, which is a regulatory domain, right? So something's going to happen to this domain that controls whether this thing can do its job. And its job is to move chloride. It is a chloride channel. It's not a pump. It's in the pump family because of the homology of the sequence to the rest of the family, but it does not function as a pump. It is a chloride channel. And here's how it works. And this informs you of why cystic fibrosis is as bad as it is. Okay, so here's your airway, trachea. And so at this epithelial level, what's going on is you're going to take chloride from the blood, and we can move it across using band 3. And band 3 is not shown here, but that's how it gets across the basolateral membrane. So this is the apical membrane. There's the tight junctions that we talked about last time. This is the basolateral because it's on the lateral side and the basal side. This side is what faces the blood. So chloride can go into the cell and it builds up an electrochemical gradient. And so if you open the CFTR, chloride will leave because it's higher, its electrochemical gradient is pointing outside the cell. And what's the charge outside of most cells? So what's the membrane potential of Joe Blow's cell? Negative 60 on the inside. So it wants to expel negative ions. And so it's getting a free ride in that direction if it just has a little bit of chloride and any kind of positive charge on the outside. Typically we say the outside is 0. That's taken to be 0. The inside is negative. So you can think of those negative charges repelling the anion, that's what you call negative ions, obviously, to the outside. It's repelling them. But you have to give it a pathway, right? And the reason for that, you now know, if you didn't already, is this lipid bilayer is impermeable to ions. It has to have a protein conductance. This is called the conductance. Okay, so if you can get this out, then everything else is going to follow naturally. What happens is you create these chlorides go out here, and so that causes the membrane potential to become more negative. It was positive, and that's going to draw sodium across the tight junctions. Remember I said some of these junctions are very, very tight. Some are leaky. This one's kind of leaky, and it's leaky to only one thing, and that's sodium. So by ejecting these chlorides, you build up some negative charge that pulls the sodium, and now you have sodium chloride in your lumen. Guess where water's going to go? If you open a water channel, the water will go into your trachea and it'll sweep away all the mucus that's building up that was secreted to trap bacteria and other things. Okay, so the idea is to keep sweeping that mucus and you get rid of it. A person with cystic fibrosis does not have that option because they cannot transport chloride, so therefore that fluid transport doesn't occur. All the bacteria stay right in this mucus layer and cause inflammation and swell the airways, it gets plugged up. I mean, one of the therapies is to hang that person upside down to get it to go. And it's not just in the airways, but that's the most critical because they just have immense trouble breathing. It's in the GI system. The GI tract transports a lot of sodium chloride, so there's gastrointestinal problems with that person, and also their sweat ducts cannot reabsorb sodium chloride, so they lose salt. And so it's not a good situation. Okay. And what's really bad is, and it's really, the most common mutation is a phenylalanine at position 508, somewhere around here. And what that does, that mutation, it causes, I think, about 70% of the cystic fibrosis cases. When that thing is mutated, what happens is this doesn't fold properly. And what happens is your endoplasmic reticulum, as you'll see very soon, is the guardian of misfolded proteins. And if it misfolds, the ER is going to eject it back into the cytoplasm, out of the membrane, and destroy it. It'll go to the proteasome and be chewed up as a destroyed protein. So it makes a perfectly good protein, it just can't get to the plasma membrane. What's really sad is if you could chill that person down to 15 degrees, it makes it. It transports it, but nobody can live at 15 degrees. So it's kind of odd. Okay, any questions about ABC transporters, about the poster children? Okay, good. All right. Oh, okay. Let's take a break here. Okay, we just have a little bit to finish up with this cystic fibrosis thing, and then we're going to go. We'll do a couple of practice problems. All right, so this is the machine. This is the CFTR, and then so we really haven't talked about it. So a lot of these ion channels are gated. That is, the ion channel is not always open. It needs to be gated. So something is controlling whether it's open or closed. We call that the gate, the gating mechanism. Okay, so that's what that little weird-looking blob is in the cytoplasm. It's the regulatory subunit, and what it's doing is it's inhibiting the ability of the CFTR to bind to ATP. Okay, so how do you unlock this thing? Well, what happens is when the cell gets a signal that, oh, we need to transport fluid and salt across a membrane, there's an intracellular molecule that goes up. It's a signaling device, and we're going to talk about signal transduction here in a few weeks. But a classic one that you should be familiar with is called cyclic AMP. The 5' and the 3' hydroxyls of a single nucleotide are sort of, there's a diester bond that causes it to be cyclic, and so it has that name, cyclic adenosine monophosphate. Okay, and so that's not an energy molecule, that's a signaling molecule. And what it's going to do is activate this protein here. PKA stands for protein kinase A. Protein kinase A. And it does the same thing that all protein kinases does, is it takes ATP, rips off a phosphate, and then puts it on a protein. Okay? And what's the protein that it phosphorylates? The regulatory domain of CFTR. That's its target. So cyclic AMP goes up, and it activates PKA, and then PKA phosphorylates the regulatory domain, which changes its conformation, and it displaces from the nucleotide binding domains, allowing them to bind to ATP. And when that happens, it splits ATP, and that's what gates the channel. Okay, so the gating is the splitting of ATP by standard, ABC transporter mechanism, and then that opens this. Instead of ejecting a drug, it opens an ion channel, and chloride leaves the cell down an electrochemical gradient. Right. So this is how you turn a pump into an ion channel. Okay, but we'll get to the how do you actually increase that signal. That's a different story. We don't want to worry about it now. The key thing is how does this channel open? That's why I give you this little piece. Okay, any questions about how it works? Okay. All right, so let's actually do some practice questions. And so pull out your clickers. And this is off of last fall's exam. So it's not just I don't think I can make that much bigger. Okay. So, answer this. So we're going to answer number 5. Start with number 5, and don't talk to your neighbors because you'll get a chance to do that. But I want an answer from the clicker first. And if you have to get up and move closer, that's fine. I'm going to try to make it just a little bit bigger. Let's see. Yeah, maybe that's about as big as I can get it. So remember, you're looking for the thing that's not a reasonable explanation. If it's a possible explanation, that's reasonable. Yes?
It's not letting us choose the answer E.
Oh, it's not? Oh, I'm sorry. I think it's going to start over. So it's not answer E. Now you're down to 2 because there's like those 2. And it was a joke. Anyway, they don't. If you chose E, you don't know what's going on.
I misread
It's really easy to get the wrong answer by misreading the question. So that's why you should be careful and read these things. Okay, everybody needs to answer. And I'm going to put, yes.
What channel are we on?
I'm sorry? 41. Yeah. Put you on the clock to get you to go. Okay, so we are down to 2 possible answers, and we're sort of equally, I like this, it's equally divided between answer 2 and answer 3. Okay, so let's go see what that is. And let's get rid of this guy. Okay, so to answer this question, you can tell now you're going to have to start using your knowledge of your poster children, of the conditions that are given in the little paragraph, and your knowledge of ways to measure membrane fluidity, right? Okay, so this animal lives where? It's in the Alaskan seawater. What's the temperature there?
Cold.
It's cold. That's all you have to know. Where do we live? 37 degrees, right? All right, so that's the key there. And so the piece of information is the human sea sark, and you know what that's our poster child for several things, but now it's a poster child for what? Membrane, type of membrane protein. It's integral and it is lipid anchored, right? So you need to know that to be able to answer this. So we're looking for something that is not, it couldn't possibly be the answer, okay? So salmon plasma membranes have phospholipids enriched in shorter fatty acids compared to those in human membranes. You're trying to explain why the human sea sark is slower in the frat recovery. That means it's the membrane fluidity of the human and its effect on sea sark is slowing it down. Okay, and the reason that could be is that the fatty acid tails are shorter. That would make the fish membranes more fluid, and that's what you need to do if you live in cold water, is have a lot of short fatty acids in fluid membranes. So that's a good explanation, so that can't be the right answer. And we know that D is not the right answer because it's this answer here, and that we threw this out. Salmon plasma membranes likely have phospholipids enriched in unsaturated fatty acyl tails. That's another way of making membranes fluid, and that's something that the salmon does do. That's why we eat fish oil, is because they use that to make their membranes very fluid, and it helps us too. Okay, so we know it's A, it's not A, and D and E, so it is between these two. And so let's look at both of these. Salmon sea sark is more likely to associate with lipid rafts than is the human. What if the salmon sea sark was all collected in a lipid raft? Would that speed it up or slow it down? Let's slow it down. That cannot be the answer. So that is the answer to this question. All right. Why is C not the answer? Salmon sea sark has a lower molecular weight than does human. Yeah, so it's faster, and that's a good explanation, but you'd have to prove that, right? Okay, good. So that's how you do these. So let's do one more. So this one you won't have to read anymore. Okay. All right. What happened to my thing? Oh, okay, there it is. Closed. Let's make it 5 answers this time. I hope it did 5 answers. Yeah.
It's not letting me choose the fifth answer.
No.
It's not letting me choose the answer E.
Oh, okay, let's try this. I don't know why, I thought I took care of that, but let's just, you can all, don't say it. All right. Yeah. Where's polling? Anywhere? No. Okay, let's start over. 5 answers. Okay. There you go. Now can you choose E? Okay, good. Okay, we're getting pretty close, so I'm gonna put you on the clock. Choose something or you'll be counted absent. And I forgot to do the group stuff, so let's do this individual, then I'm gonna let you talk to your group. Okay, everybody thinks it's answer 5. No, not everybody, but, all right. Save. Okay, yeah, whatever. Put it back up. So turn to your group and then work on it while I figure out what's going on here. Sorry.
I feel like it's just like a question, but it's not what it's not I put E because right now
So one person in your group is clicking in the answers.
A is
I don't really think that's I think it's
Okay. At 15, we need 3 more groups to click in. That's on everybody. Good. Okay. Wow. Okay. So, all right, get out. All right, now. All right. So, to answer the question, you had to know stuff about FRAP. You had to know what an incomplete recovery indicates. You had to know the structure of SARC. And so let's go through this. We're looking for the thing that couldn't possibly be an explanation of this. If it is a possible explanation, then that's not the answer to this question. So you got to get that part straight. And you got to understand what's going on. It's the human thing only recovers 60% of its fluorescence. What does that mean? 60% of the SARC is mobile, and the space between 100 and 60 is the immobile fraction. So anything that jives with that is not the answer to this question. So 60% of human SARC is free to diffuse freely in the membrane, whereas 100% of salmon sea SARC is. And that's because salmon sea SARC recovers 100%. So that's perfect. That's not the answer. About 40% of human sea SARC binds to a single pass membrane and that are immobile, whereas salmon does not. Okay, is that a good explanation? Yeah, that's a good explanation. So that's not the answer. Okay. About 40% of human sea SARC binds to extracellular matrix proteins like collagen, whereas salmon sea SARC does not. Would that keep it immobile? Is it 40%? Yeah. The problem is, if you know the structure and where SARC operates, it can't possibly bind to anything in the extracellular matrix because it's a cytoplasmic protein. That's the answer to this question. It has to do with your understanding of which side of the membrane sea SARC is. That's not a trick question. That's your knowledge of sea SARC. Yes.
So, the extracellular matrix proteins like collagen, are they not integral proteins? Do they not have some minimal cytoplasmic domain?
Not collagen. It's a completely extracellular matrix protein. It does not go through the membrane. Are there receptors on the plasma membrane? You would have to know that. I mean, I would have to give you an example where it's a see, like I said, it's a single pass membrane protein. I would be leaving out critical information if I did. But collagen it's actually this matrix that the cell is actually grabbing onto using transmembrane protein. Sea SARC is not a transmembrane protein. Is it integral? Absolutely. It's lipid anchored on the inside. Are there lipid anchored membrane proteins that are extracellular that actually would fit this? What's a good example? There are three. Well, glycoform's a transmembrane. So a lipid anchored extracellular matrix, I mean not extra, but has a domain in the extracellular causes bad cow disease. Prions. It's a GPI anchored protein. That's one that if I'd given you that thing, then that would be, then C would be a reasonable answer, and that would not be the answer to the question. Okay. Okay, so let's proceed. Okay, so we're starting to be able to, we've got a lot of players now. Now we can start to put them together to understand this learning goal. Predict the consequences of placing transport proteins in different locations in the cell membrane. Okay, and so we're good. A good way to answer that type of question is to look at epithelia. Now, we've already looked at the pulmonary, or not pulmonary, but the tracheal epithelia, right? So we can do that with other tissues. This is just to remind us that, and we've already gone over this point, that remember that ion gradients represent if there's a difference, an electrochemical difference, they represent a stored form of energy, okay? And so you don't have to have ATP being split by the transport protein. You can actually use an ion gradient to couple that to the movement of another solute against its electrochemical gradient. So we're going to start talking about coupled transport. Okay, so the coupled transport is all secondary active. Everything we've been talking about is primary active because it splits ATP. Secondary active proteins work off of ion gradients. That's where they get this. So they capture that energy and use that to push something else against its electrochemical gradient. If you're a uniport, you can't do that. You need to have two different solutes. Not stoichiometry of two different ions, but they have to be different species. Okay? So symport is when those two different molecules go in the same direction. Antiport is when they're exchanged, like band 3, bicarbonate for chloride. Okay. But those are both coupled types of transport. They're both secondary active. Okay. So here's a really bad cartoon, but we'll look at it anyway. So this is the sodium glucose transporter. And so its job is to capture sodium and to capture these little yellow glucose. You didn't know that glucose is yellow, did you? So it's got to capture the glucose and the sodium before it is allowed to turn and change its conformation. It's an alternating conformation model, right? But what makes it go is the fact that Sodium has an inwardly directed electrochemical gradient. There's energy there. More sodium, less sodium on the inside. Why is sodium low on the inside of all cells? Because of the sodium-potassium pump. See, now you're starting to think about different pumps working together, different transporters. Okay, so the ATP, ultimately, the energy for the sodium gradient is coming from ATP because of the sodium pump, right? Now, this guy is going to use it to bring in sodium, but also glucose. And the stoichiometry is wrong here. What it does is it binds 2 sodium ions plus 1 glucose, and then flips it across this membrane. And the person that cloned that is Ernest Wright, and I worked across from his laboratory for 5 years at UCLA Medical School, and he came here last fall as the honored speaker at the medical school. And so my whole class got credit for going over there and meeting Dr. Wright. But he's the one that figured out the structure of SGLT1, sodium glucose transporter. But see, here's the sequence. Here's the open. This would be the so-called T1, transporter 1 conformation facing outside the cell. And this thing is always on the plasma membrane, by the way. Okay? It binds 2 sodiums, and then you load on 1 glucose. And when everybody's situated, it now refolds. And the binding sites now face the inside of the cell. And you lose the sodium, you lose the glucose, and then you get to do what you want with that. Okay? And what you're going to do with it is you're going to transport it into the bloodstream, because the cell type that has this almost exclusively, there's one exception, is your gastrointestinal epithelium. So your enterocytes, which is your absorptive cells, are going to use, that's the reason you can absorb glucose. And it is actively absorbing every little smidgen. Because if you leave behind glucose into the GI lumen, you get diarrhea. And that sounds funny, but it's not to people that have it. Because there are people, right, we all know that. And then there's familial diseases that are based on the fact that this thing is mutated. So if you get a newborn and that newborn cannot, it has intractable diarrhea, they either have cholera or short bowel syndrome, but this is a possibility. It's rare, and a lot of your colleagues are not going to know this, that this is a possibility. All you have to do is take them off of mother's milk, because lactose is the major sugar in mother's milk, correct? And lactose is a disaccharide of glucose and galactose. Glucose and galactose are both transported by this thing. So if you give them fruit sugar, fructose, they don't have diarrhea anymore. They don't die. So it's important to know these possibilities. Okay, so now let's put them into context. This is an enterocyte. We've already looked at the junctions and all that stuff. This is the Bart Simpson haircut, right? And these are called microvilli. And the reason they have a little spiky haircut is because it's standing room only. There's so many proteins. It's not just to give the absorptive surface area a boost. That's one reason. It's because you can't fit any more proteins in there. There's one transporter for almost every class of protein, and this is how you get fed. So the nutrients are coming in here. Let's say you did ingest lactose. That gets split into glucose and galactose. They're both transported by SGLT1. And you put SGLT1 right here on the apical membrane. Okay? Now, glucose cannot go across the junctions because we want it to go through the cell. And so glucose comes into the cell, and then glucose needs to exit the cell. And so it's going to exit on the basolateral membrane. And to do that, we use a different protein. We use an old friend of ours, GLUT2. What do you remember about GLUT2, by the way? You remember that sequence we did GLUT1, GLUT2, GLUT3, GLUT4. GLUT1's the one that's on everybody's cells, okay, but not here. We use a different transport. Remember, it's the first thing that sees glucose, so it's going to see the highest concentrations of glucose. It is a really sloppy KM, about 13 millimolar. And that's the reason it can get by with a sloppy affinity, is because the SGLT1 transporter is building up the concentration of glucose, and then it's going to leave down its concentration gradient. So now you have a way of transepithelial glucose transport from the gut lumen into the bloodstream, and that's how it does. And then this is going to drag water too, because how do you eject the sodium that's coming in? You want to keep it low, right? To keep the sodium sucking in, and so you have to shoot the sodium outside using the pump. So if you put this there, this system is not going to work, is it? Think about that. What's going to happen is you'll bring in glucose, all right, but you won't bring in sodium. You'll bring in sodium, but you're going to kick it right back out. You just recycle sodium, and so that's going to cut down on fluid absorption, and you're still going to get diarrhea because you can't absorb the water. So it's important. If you put this thing here, would it work? No, it wouldn't work. What if you put GLUT2 on the apical membrane? Well, it would work, if you just had eaten a candy bar and your luminal glucose was higher than in here, but after a while it's going to decrease below the Km of GLUT2, and that glucose is going to stay in the lumen and you get diarrhea again. Okay, so you need something that mops up everything, and that's why nature put it there. Okay, does that make sense? Okay. Okay, and this is, what does this show us? Oh, it just shows that the junctional complex here, that's the tight junction, and that's why the sodium and glucose and water can't go across there. It's forced to go through the cell so that we can regulate it. Okay. I don't think we're there yet. All right, let's go back to the outline. So this is our first example of a coupled transporter. This is a symport, correct? Because it's going in the same direction. Oh, one thing I did want to say, and before we switch over, is there is another isoform of this transporter. This is called SGLT1. Let's go back to the model. This is called SGLT1, and they subsequently, and so there's only two places where you find that. You find it in the GI epithelium, and you find it in the nephron, the kidney. Because what happens is when your blood is filtered by the kidney, all the glucose in your blood is taken out, and it goes into the nephron. And if you leave it there, you're going to pee it out, and you'll lose all your glucose. So the nephron is designed to reabsorb all of that glucose back into the bloodstream. And we used to think that this is the only thing that the kidney had to reabsorb glucose back in the bloodstream. It turns out that its friend, SGLT2, is the isoform that's present in the nephron. And the reason I bring it up is because it's extremely medically important. It's actually a new treatment for diabetes. So let's see how we can get all that figured out. This is probably the last thing we can talk about today, but let's go ahead. And this is something that Dr. Wright talked about in his lecture to the medical school. And so you need to know just a little bit of information about the nephron. So you have hundreds of these little filtration units. It's called the nephron. And so your blood's coming in, let's make it red, right? And it breaks up into a little ball of cells, a little capillary ball called a glomerulus. There. And that's where all the glucose comes out of the blood into this thing. There's a cup, it's called Bowman's capsule, that surrounds this. And so this is Bowman's capsule. And so all that stuff is filtered, all the little things. The proteins cannot be filtered by the nephron. If you get protein in your urine, it means you have nephritis. It means you're losing your kidney's ability. That's a really bad deal, okay? But the little things like amino acids and glucose and nutrients are going to go into the lumen of this little filtering system. And the first thing it goes into is called the proximal convoluted tubule. And you notice I didn't draw it as a convoluted tubule, but it is. It's all kind of twisty to get more surface area. And then it breaks up into what's called the loop of Henle. And this is a really thin tube that goes up, and it comes back down, and then it gets kind of thick. And we don't need to know the anatomy of the rest of it, because glucose is all absorbed back into the bloodstream really quickly, because you don't want to lose it. So this is the loop of Henle. Okay, and where is the SGLT1 and 2? Well, it turns out that, let's put color. SGLT, there's about 10% of your glucose gets reabsorbed right around here in the descending part of the loop of Henle. So that's 10% is due to SGLT1. And we used to think that's the only thing that was there. But it's actually wrong. The main protein is actually in the proximal tubule, and that's SGLT2. It actually has a lower affinity for glucose, but there's lots of monkeys in the room. It's like an enzyme that's moving nutrients. Each monkey is not very good about pulling glucose back in the bloodstream, but there's a lot of monkeys. So it's high capacity, low affinity protein. That's what high capacity means. There's lots of them. Okay? So that's how 90%, let's just pull this up, 90% of your glucose gets back into the bloodstream. And so by the time it comes out here and it goes out and you form urine, all the good stuff has been removed. The amino acids, the glucose, all the nice ions, and you just get rid of the liquid that you don't want. And there's a really elegant way that the kidney does that, but that would be an entire course to talk about that. Okay, so where is this information going to lead to that makes a drug for diabetes? Well, it turns out that we've known for some time that a really good inhibitor, and this is where your toolkit comes in, there's a drug called Florazin. Okay? And what that does is it blocks SGLT1 with high affinity. It also blocks SGLT2, but it's not very good with SGLT2. So it's a weak inhibitor of SGLT2. So what the pharmaceutical companies said, okay, and the concentration of SGLT2 is determining how much glucose goes back into your bloodstream. What if you're diabetic? What do you want to do with the glucose? You'd actually like to pee it out, right? And so what happens is, let's block a little bit of SGLT2 because it sets the amount of glucose returning into the blood. And if we just turn it down a little bit, you don't want to kill it, you want to turn it down, take a little bit of this reversible inhibitor, the glucose you actually release in the urine, and your blood glucose is clamped at a lower level, whether or not you take insulin. That's amazing, because if you have to take insulin, it puts weight onto people, and it's a difficult thing to deal with. A lot of type 2s are taking pills that making their insulin production higher, but usually it's not enough. And so they still have elevated blood glucose. So if they go on to insulin, they're going to gain weight, and that's not good. This thing is an insulin-independent way of excreting glucose. And so what they did, the pharmaceutical companies took a look at this structure and they started fooling with it, and they came up with this thing, dapagliflozin. And you can think of flozin as flowing, because it accelerates the glucose to flow out. That's the way I remember this thing. Okay? It's an exquisite inhibitor of only SGLT2. Right? So it's 1,200 times better than Florazin. Why would you want to make it specific for SGLT2 and not for SGLT1? SGLT1 is transporting a little bit of that glucose. You take that pill, what's going to happen to you? It'll work on the kidney. It's going to hit the one where? Your gut, you will die from diarrhea. You can't do that. So, well, you can, but you lose the patient. But, so you take this thing, it's insulin independent, it lowers blood glucose and doesn't put weight on the patient. And so these are coming on the market fairly soon. Really brilliant. Okay, any questions? I didn't mean to teach you so much renal stuff, but now we're getting to a position we can talk about these things that matter clinically and play around with the different drugs, and so all this stuff is happening. It's really exciting area. Well, we'll just stop there today.